Hello everyone, this is lecture 5 of course BE544. In today's lecture, we will deal with different terminologies related to artificial intelligence, machine learning, and deep learning, and then go through different codes to handle the annotations and whole slide images, how to extract the different tiles from the image using Python. So let's begin. As we mentioned earlier, the course title contains artificial intelligence or AI, and simply, artificial intelligence is how to develop a software or a program or a device that can sense, reason, act, and adapt to the environment based on set of rules or patterns. And machine learning is a subset of artificial intelligence that contain different algorithms that can learn from the input data and create different patterns that can mimic the human brain and how the human brain is thinking. Machine learning can be supervised, unsupervised, and reinforcement. Supervised when you are providing the model with the input data and outputs, so the model can learn how to map or drive from the input to the output. And unsupervised learning, where you are only providing the input data only to the model, so the model can learn from only the input data and try to drive some patterns or clusters that can group each of these input data or records. And reinforcement simply is learning from the environment by sensing different features and inputs from that environment and learn from it. Deep learning is a subset of machine learning, which basically is established on the idea of neuron that mimic the human brain. So the word deep here in deep learning basically means you are developing a complex model or a complex system that can handle complex data sets and large amounts of records using different techniques such as convolutional neural network CNN or state-of-the-art models such as vision transformers. So in that course, we are focusing mainly on the idea of supervised learning in machine learning. So we have input data and outputs, and we are trying to guide the model or the system to get patterns or maps to map from the input to the output. We are going to deal with different algorithms of machine learning, including deep learning models or algorithms. So as you see in the right figure, there is a whole slide image with annotated region in red in the middle of it called tumor. So if you are going to apply the idea of supervised learning on that whole slide image only, so we can simply say that only the annotated region is tumor, but the outer region is not tumor, which can be considered normal or benign. So we now have two regions. One is considered tumor and the other is considered benign or non-tumor. So if you are going to use a supervised machine learning model, we are providing that model with two things to learn from them. The first is the tiles or the images and the corresponding class or category for each image. For example, we can provide that region to the model with a corresponding class of tumor. We can also provide the model with that region with a specific class called non-tumor. And by providing these images with the corresponding classes or categories to that model, it will learn to map from the input data to the output. So simply, in the testing phase or the production phase, we can ask the model about these regions. So it will take this input data and based on the learned features and learned patterns, it will say it's non-tumor or tumor. So our aim or mission is to find and tune the model to accurately and precisely map from the input data to the output. So we have multiple models, many ideas, many techniques, but we cannot guarantee that all of them will work with a specific dataset. So we are trying different datasets with different configurations, and we are aiming to tune one of them, aiming to reach high performance such as high accuracy and high precision. We now understand what is the meaning of classification. So classification here identifies the category or class in which an object in an image belongs. So as you see here in this picture, we can say this belongs to a cat, and in our example we can say it's a tumor or non-tumor. Other applications of machine learning and deep learning can include localization. Localization means is to determine the specific location of an object within an image, and this is simply by providing the coordinates of it. The coordinates here means x, y, the left upper bound, and width and height of the boundaries. By combining classification and localization, as you see in the second image, we can say there is a cat which is located in the coordinates x and y with a width w and height h. So if you combine both classification and localization to identify and locate multiple objects in an image, this is called object detection. So you can say that image can include a cat and a dog. The cat is located in that place and the dog is located in that place. But if you divided the image into specific or smaller segments and labeled each pixel according to the object or region it belongs to, this is called segmentation. So you are trying to extract the specific object from it instead of setting a boundary or a box that surrounds that object. So if you see the difference between object detection in the third figure and segmentation in the fourth figure, you will find that detection only sets a boundary to the object, while segmentation extracts the exact object from the image. So segmentation goes beyond object detection. This is done by creating boundaries around each object instead of just introducing bounding boxes. There are two types of segmentation: semantic segmentation and instance segmentation. Semantic segmentation labels every pixel in the image according to a class. So if you see in this figure, you will see that it labeled the road with a gray, the grass with green, and all sheeps with the orange color. But instance segmentation advances semantic segmentation by differentiating between the different instances that belong to the same class by assigning a unique instance ID to each of them. So by doing that, it will separate between each of the objects. For example, sheep 1, sheep 2, sheep 3 can be split from the image without considering that each of them belong to the same class. So now we understand the meaning of classification, localization, detection, and segmentation. And as we said, not all of the algorithms will work with all of the datasets, and we need to tune and carefully select which algorithm to use, which technique to apply, and which configurations to handle. So what we see in this slide is a published classification workflow using whole slide images by simply including only the histopathology region and ignoring the outer region, because the outer region does not belong to the histopathology slide, and then extracted tiles or batches from the whole slide image and categorized each of these whole slide images. For example, the first is class A, the second is class B, and so on, and then applied these tiles or batches to a model and get the results after that. They have used a neural network to train these batches. So a neural network is an approach or a technique in machine learning which is based on the concept of neurons, and we are going to deal with that architecture in the future lectures. But the idea here, they didn't use segmentation, they didn't use localization, and so on. And this means you don't have to use all of the techniques or all of the approaches in your application. It depends what is the aim of the project, what is the problem you are going to solve, and what is the target or the objective. So in this course, you are getting different techniques with different algorithms, and you need to specify the best approach to solve your problem. This is another workflow from another published study. They only extracted the annotated parts, as you see in the left figure, which are in green, and then applied on the extracted region of interest their model, which is a deep learning model, and produced the results. This is a third approach by simply capturing the idea of the pyramidal view, by simply extracting tiles from each of the magnification levels and introducing these tiles to a separated model, and then finally combining all of them together to get the output. So for example, they extracted a tile at 25x and the corresponding tile at 100 and the corresponding tile at 400, and then applied the three of them to three separate models. VGG16 here is a pre-trained deep learning model that we are going also to deal with, but here we are only getting the idea, and then they will get three different vectors which is concatenated together to get the final output. This is also an approach to solve your problem. So as you see in the three workflows and many other flows, you will find that the first step is the pre-processing, which focuses on extracting the tiles, whether it's from a single pyramidal level or multiple pyramidal levels. But the idea here is to extract tiles from the whole slide image. The reason is that you cannot handle the whole slide image as a whole with a model, because it will consume much resources and may not fit to that resources. For example, if you are dealing with whole slide images with 100K by 100K width and height respectively, it will be hard to find a device that can handle the whole slide image as a whole. So you need to extract patches or tiles from it. Second, the whole slide images may not belong to a specific category, such as tumor, but the whole slide image can include different categories such as benign, malignant, carcinoma in situ, carcinoma invasive, as we saw in the previous lecture. So you need to carefully, based on the annotations, extract the tiles and categorize them carefully, and using these tiles or folders that contain the different tiles to pass them to the model. So as mentioned, we are going to use Python to handle that process. There is a package called OpenSlide which can be downloaded and integrated with Python to manipulate and handle any whole slide image. You can simply go to openslide.org/download. You will find different options here. Scroll down until you find something called binaries, and then download for your operating system, for example Windows. Try to download the latest version and extract it in any region or location, but it's recommended to extract it in the Windows partition, for example C. As you see here, OpenSlide version 4.003. If you open that, ensure that there is a folder called bin and lib, which reflects binary and library, and then open the command window using any approach. For example, you can open run and then write cmd to open the terminal, and then you need to install the OpenSlide package using the pip command. So you write pip install OpenSlide-Python, and this requires that you have installed Python or you have an already installed Python version. And throughout this course we are going to use Python version 3, and as you see here, it's installed as we have previously installed this package. And to ensure that you have installed the package successfully, simply open Python and then write import OpenSlide. It may get an error like that cannot locate OpenSlide DLL, but we are going to handle that issue inside the code. But until that point, this means we have installed the OpenSlide package. Throughout the different codes in this lecture, we are going to deal with that file with the corresponding annotation. It's called AO2, SCVS, and XML files. If we simply open it, you will find the different annotations such as benign, carcinoma in situ, and carcinoma invasive. And the non-annotated region here belongs to the normal. So we have 4 classes. As we got here, we got an error that we couldn't locate the OpenSlide DLL file, and we said we are going to handle it through the code. So if you went to the installation of the OpenSlide in the official website, you will find there is a quick fix to install OpenSlide on Windows. So simply you will copy that at the beginning of your script and just modify the path of the extraction of the OpenSlide. As we said, it's recommended to extract it in the C partition. So you go to the bin directory and then you copy that path and then you paste it here like that, and ensure that the R letter prefixes the string. R here regex, and simply the usage of R here is to inform the Python interpreter to deal with the special characters as raw characters or normal characters. For example, \n in the programming language means new line. So if you wrote here \n with the letter R, this means deal that \n as just two individual characters rather than dealing with them as a special character. So the R character that prefixes the strings here, especially with the paths, is recommended. Then you need to import the different packages such as OpenSlide, which we installed using the pip command, and matplotlib for visualization. And if you don't have the matplotlib in Python, you can install it using pip install matplotlib. Then we have specified the path of the SVS file, and if you look here, we didn't specify the absolute path of it because the file we are going to use and the code are in the same directory, so we can use relative paths. So update that to your SVS file you are going to use. For example, we are going to use AO2, so let's change this to AO2. Then you will create an instance from the class OpenSlide in the OpenSlide package by pathing the path of the SVS file, and this will create an object. Let's call it slide. This object will contain different functions and properties that is related to that OpenSlide file or SVS file. We can access the different properties of that slide, such as dimensions, which is the width and the height, the number of levels, for example 3 levels or 4 pyramidal levels and so on, and the dimension of each level. So AO2 contains 3 levels, so we can access the different levels using the list, level dimensions, and pathing the index of that level. 0 is the highest magnification, and by increasing the index it will get to the upper levels of the pyramid. So if you executed that part only by running the code, Python, and the name of the file, and ensure if your file or Python script contains spaces, you need to write it between double quotes. You will find that the dimensions is 54K by 46K in width and height respectively, and it contains 3 levels, each levels with the corresponding dimensions. For example, this is the highest magnification level, and once you get to the higher levels of the pyramid, you are getting narrower and less of details. As you see here in the top level, you have only 3K by about 2.8K. Let's remove that, and you can display a region of that slide using the function readRegion by specifying the XY top left corner of the region and which level you need to display and width and height of that region you need to display. And for sure, if you said you are going to display 54K by 46K in the level 0 or the first level, it may hang your device because it will require much resources such as RAM or memory to handle that. So we are going only to display a patch or a tile with a size 1000 by 1000 from the first level. So you are creating a figure and displaying the tile and turning off the axes and removing any unnecessary padding or margins, and then displaying that figure. If you executed that code again, It will display the same details and present the tile from the coordinates we specified with a width and height 1k by 1k. So for example, if we set at the endpoint, which is on the right bottom corner, the x and y will be 1k by 1k. And if you look here, while going through each pixel, there are 4 values that are displayed in this region. Look again, we have 4 values in a list, where the last value is 255. These 4 values represent the RGBA color. R is for red, G for green, P for blue, A is for alpha, which is a transparency. So all of these pixels is 255. And we are mentioning this point here because we will require to remove that channel later in the processing phase, as we are going to see in the next few codes. You can also change the level from 0 to 1 and rerun the code. It displays the region with 1k by 1k, but you may say that we have mentioned 20k by 20k, which is the left upper corner of the starting point that we need to capture the region, which should be considered that point in the second level or level 1, and we have only 13k by 11k in width and height for level 1 or the second level, which you may say this is much higher than the width and the height. You are correct, but the red region here, or this function, requires x and y in level 0, and it internally will handle and map from the first level to the second level to the third level and so on. So xy here are for the first level or level 0, and you specify the level, and this function will handle the mapping by finding the corresponding coordinates in level 1 or level 2 as you are specifying here, and then capture 1k by 1k. So if you specified level 2 and we have only 3k by 3k approximately, and you are specifying start from point 20k and 20k, as we said, it will internally handle the mapping. So as you see here, it have displayed 1k by 1k at these xy coordinates specified at level 0 and mapped to level 2 or the third level. So in that code, we are able to read the whole slide image, get the different properties from it, such as the level counts and the dimension for each level, and then displaying a tile or a portion from any levels that we need. What about the annotations? We can read the annotations using the XML built-in package in Python by specifying the XML file and start to define the hierarchy of that annotation to Python. If you open the annotation, which is ao2.xml file in any editor, such as Sublime that I'm using currently in this video, you will find that there is a hierarchy. And if you are familiar with XML file, it consists of tags and properties, where the tag can be a parent that contain set of children, and each tag can be opened or closed like that does not have any children, or open like that or like that can contain children inside it. Simply to differentiate between open and closed tags, you simply look forward for the open tag and its closing. For example, the tag vertices here consists of different children, and then at a specific point you will find the same tag but with a prefixed forward slash. This means this is an open close tag that include children inside it. But the open tags or self-closing tags like that one has a postfixed forward slash at the end of it, and similar to that, this means this one or this tag cannot include children in that format. Self-closing tags can be used to define attributes or properties, while open closing tags can include properties and the children inside it. And this can be like a family. This is the grandparent, this is the parent, and the parent have children, and each child can have also children, and so on. So to access, for example, the regions, we need to specify the first tag, and then from this tag we need to search for the regions tag, and to search for the vertices we need to access the annotation, and then regions, and then vertices, and so on. As you see here, when we collapse the vertices, you will find that there are different regions. This is the first region that contains vertices, this is the second region that contains vertices, and so on, and all of these regions inside a tag called regions. And if we collapse it, you will find that all of the regions are included in the annotation, and this is only one annotation. It can be multiple annotations depending on the file or the annotation behavior, and all of them are included in the root tag, which is called annotations. Why do we see that? Because you need to understand the hierarchy of the XML or the annotation file so that in the programming you can handle it correctly. And this hierarchy differs from a dataset to another based on the annotator, type of annotations, type of classes, and so on. So based on the dataset you are going to use, you need to look through the dataset annotations and define the hierarchy, and based on that hierarchy you need to write your code. So in our code we have imported the XML built-in function and then matplotlib for visualization, as we are going to see, and defined the XML file which is called ao2.xml, and the type of definition here is relative because we are setting the code and the file in the same directory. We first start to parse that XML. Parse here means translate or interpret that XML file as an object. And if you executed that code or that line and got an error, this means the library couldn't parse it, and this means there is a typo or a mistake in that file, or someone manipulated it in a way that cannot be parsed using that library. After that, we got the root object by the function getRoot, and the root here, as we highlighted in that file, is the annotations tag. So if we simply printed the tag of the root by writing root.tag, tag here is a property that prints the tag name of any tag. It's not necessary to be the root. We can write for any of the nodes .tag to get the tag name. You will find, as we mentioned, the annotations. So we need to find all internal annotation tags inside the parent annotations tag, because we mentioned it can include different annotation tags, but in the file we opened it only included one. So just in case, we need to find all annotation tags and loop through each of them. How to do that? We simply use the root and use the function findAll and pass the tag name by passing dot to forward slashes and then the annotation tag. So it will find all annotations. If we printed the length of it by simply printing number of annotations, you will find that we have only one annotation, as we saw in the XML file. So by looping on each annotation of the annotations tag, we can access the inner tags of each annotation. So as we saw, we have a single annotation, and inside each of them you will find that there is a single annotation with regions inside it. So we need to loop on each region of these regions, and inside each region you will find another tag called region, and in each of the region tags you will find vertices. So we need to go through that hierarchy. So for each annotation we have accessed all regions by the region tag and printed the number of regions. So for example, if executed only that code or that loop, it will print that we have a single annotation as mentioned, and inside it there are 17 regions. So we need to loop on each region of these regions, and then we get the vertices. So each region contains vertices, as we are accessing here. What about that line? If you remember in the image scope lecture, during the annotation we mentioned that there is something called text and there is something called description, where we can write text or a class or a category in the text and the description in the description field. So we are accessing the attribute called text. Why? Because if you went through exploring that XML file, that you will find that the category of the region inside the text. For example, the first region is benign, the second region is benign, the third is benign, and for the one with an ID 6 is sarcoma in situ, and so on. So we can get the class of that region by accessing the attribute. And you need to understand carefully that this attribute can differ from a dataset to another dataset. So you need to carefully explore the XML file before dealing with how to handle it using Python. So now we have accessed the vertices. We are printing the number of vertices in each region. So if you executed that part only, you will find that for the first region it's benign with 83 vertices. There are other regions, but we have exited the code at this point to understand the hierarchy. But if you continued to run that code, you will find the number of the other vertices in each region. So to access each vertex in the vertices, you can access using the attribute. As we mentioned here, we can access the text attribute using the attribute. So what is the benefit of vertices here? Each vertex have coordinates. By getting these coordinates, we can get a polygon that surrounds a specific region in the whole slide image, so we can easily extract that region to a corresponding or a specific category such as benign or sarcoma in situ. So how to get the XY coordinates of that? We need to open the XML file and ensure that the property is uppercase X and uppercase Y, and Z for all of them is 0, so we don't need it because it's not a 3D. So we only access X and Y from each vertex, as we mentioned here. To understand that part, this is simply related to something in Python called list comprehension, which is a shorter version of creating a new list using a for loop. So instead of creating a loop such as for each vertex in the vertices, append or add a new element to the list called chords, we simply use the list comprehension by adding two square brackets, apply what you need to process here, and then the loop. So we need to loop on each vertex in the vertices and apply that. Get the attribute X and Y of each vertex, and this name should be similar to that, and then convert it to float and then convert to integer, and this is for X and this is for Y. To get the coordinates, we are concatenating and putting both of them in a tuple. So if you printed that coordinates list, you will find it's a list of tuples where each element in the tuple is the integer value of X and the integer value of Y. As you see here, it has printed all of the vertices and each of them is a tuple, as you see here. As mentioned earlier, we have about 17 regions where each region has a polygon or set of vertices that construct a polygon that surrounds a region with a specific category. So we need to combine all of these polygon or regions in a list so we can later use them. So we have created here an empty list called annotation list, and then after constructing the coordinates, we are appending in that list a dictionary like that. That dictionary consists of two fields: text, which reflects the category or class, for example benign, and the coordinates, which is the list of coordinates that we constructed here. So if we printed the list length, which reflects the number of annotations, it should be 17, similar to the number of regions. As you see, the number of annotations is 17, and when we executed the code, it said there are 17 regions, where the first 4 are benigns, then carcinoma in situ, and so on until you reach the last one, which is carcinoma invasive. And for each, it defined the number of vertices. For example, the last one contained 837 vertices. So we now are able to handle the XML file for that dataset. We understood how to explore the different tags of any XML file and reflect that in a Python script. As we saw in the whole slide image and the annotation, there are 4 classes in this dataset, which is called BACH dataset for breast cancer. These 4 classes are normal, benign, carcinoma in situ, and carcinoma invasive. And we are going to deal with these 4 classes in the lectures. So we need to understand what is the meaning of each of them. So normal class represent healthy tissue with no abnormal cell growth or malignancy, and the cells function normally and maintain the regular structure and function of the tissue. Benign is non-cancerous growth that don't spread to other parts of the body, and there is no serious threat to health for benign cells. These benign cells may need removal if they cause symptoms or complications. Carcinoma in situ and carcinoma invasive are cancerous cells. Carcinoma in situ happens when abnormal cells are present but have not yet spread beyond the original tissue layer. It's considered an early stage of cancer and can often be treated successfully if detected early. But carcinoma invasive is a more advanced stage of cancer where abnormal cells have spread beyond the original tissue layer into surrounding tissues. This type of cancer can metastasize to other parts of the body and often requires more aggressive treatment. So by understanding these annotations and being able of reading the annotations and the whole slide image, we need in this snippet to apply the annotations on the whole slide images and draw the different boundaries on the regions and then compare what appears in our visualization with the original whole slide image in ImageScope application. So at the beginning, we have included this snippet to include the DLL file for the OpenSlide package, and then imported OpenSlide to read the whole slide image, and the XML package to read the annotation, and matplotlib for visualization. Then we defined both of SVS and XML files, and we have included both of the scripts together in a single script. First, we read the slide and made an object called slide, as we did in the first code, and we made annotation list and then read the different coordinates and the classes similar to the second code, and then we made the visualization. We need the visualization to look like something like that, where each region can take a specific color. For example, the benign can take green, the carcinoma invasive can take red, and so on. And we need to display the whole slide image as a whole. But we mentioned earlier that if we imported or loaded the 50K by 50K width and height respectively whole slide image, it can consume all of the resources and may corrupt the code or hang your PC or laptop. So we simply will get only a thumbnail using the function getThumbnail with a specific dimensions or reduced dimensions. So we read the dimensions, width and height, and divided it by 50 and 50. Then we displayed the thumbnail. If we commented this part to see what will be visualized and executed the code, it may take time according to your resources such as memory of your PC or laptop. And as you see, this is the thumbnail of the whole slide image. Let's uncomment this region. So we here have annotation list that contains dictionaries, where each dictionary consists of the class or category and coordinates. Then we looped on each dictionary here so that we can get each dictionary and the coordinates, and based on these coordinates we can draw a polygon and fill it with a specific color. We got the XY lists using list comprehension by looping on each coordinates of the coordinates, and as you see, the coordinates from the previous example, it was a tuple where the first element is the X and the second element is the Y, and here we need to extract all X's and all Y's, and here we divided by 50 because in the thumbnail we divided by 50, so we need to handle all points accordingly to each other. Then based on the text or the category, if it's benign, set the color to green. If it's a carcinoma in situ, set the color to yellow. And if it's carcinoma invasive, set the color to red. And otherwise, if it's normal, set it to blue. Then, based on the XY coordinates, we use the function fill to fill this region or this polygon with a color with a transparency of 50%. Then we plotted a boundary with the same color but no transparency, so you'll find the filled color is transparent more the boundary. And here we have added the starting point as the ending point to close the polygon. Then we use the text function to write the category of that region on the filled region with a font size of 5 with a color black, and the coordinates of that text is the minimum of X and the minimum of Y to be the left upper corner point or according to the minimum values locations. And finally we closed the SVS file to release it from the memory. If we executed that code, it will take a bit to display. So as you see here, each region is filled with a color, for example red with a transparency of 50% and outlined with the same color without any transparency. And if we opened the image scope, we will find both of them are similar, and this confirms that the code runs accurately. So in that code simply we have merged between the first and second codes by reading the whole slide images and the annotations, and combined both of them by extracting a thumbnail and applying filling and plotting based on the coordinates of each region in the annotation list. And as mentioned, you need to update the thumbnail and coordinates accordingly based on the division ratio. For example, if we set it to divide by 100, 100, 100, 100 here, we will get smaller region and the plotting will be faster because you are getting a small region or thumbnail from the object or the whole slide image. As you see here, we got a reduced size. It's half the size of the previous visualization. It was 1000 by 1000. Now it's about 500 by 500 with the same visualization but in a reduced resolution. So we use the function getThumbnail in visualization purposes, while in the first code we used the readRegion function to read a specific area with coordinates X and Y, width and height, and we define the level that we need to read from, and that function will be used later in extracting the patches or tiles. So in the next code we need to read a specific ROI or a specific region of interest based on the polygon we have filled and plotted in the previous code. So as you see here, for that region we need only to extract that benign region, and for that region we need only to extract that benign region, and so on. So how to do that? Basically we can use the polygon that we filled with red or green or blue in the previous code and fill with white, and other regions should be black. And then we can apply masking from that mask that contains white and black pixels on the region image. So by multiplying white by any color, it will produce the same color, but by multiplying zeros or black by any color, it will be zero. So this white region will only filter the benign region of or region of interest as we see in the right figure. How to do that? Simply, we have included in that code the same snippet to import the DLL package of OpenSlide, and then we imported OpenSlide, XML, Matplotlib, and here we also included OpenCV package for image manipulation and NumPy package for numerical manipulation. Then we included the SVS and the XML files, read the SVS file, and included the list of annotations as we did in the previous code. And then we have specified to work only with the first index of that list, which is the benign region, and later we can change that index to 1 or 2 or 3 according to the region that we need to extract. Then we extracted the x's and y's of the coordinates as we did in the third code. Here we have extracted the x's and y's, and then we calculated the region width and height so that when we apply the function read region, we can specify the width and height of the region to read. To get the width and height, we can get them by getting the maximum of x and the minimum of x and subtract both of them to get the width, and the same for y. The maximum is the bottom and the minimum is the upper or the top. By subtracting both of them, it will get the height. And then in that part, we wanted to get the mask, which is similar to the left part here. But let's consider something. This point here should be 0 and 0. But if you looked here, the benign region is shifted to the bottom and right, or the original place in that location. So when we need to deal with that region, we need to map the coordinates from this current coordinates to be shifted to the upper left corner. To do that, we can subtract all coordinates from the minimum values. So it will be shifted from here to here. Why are we doing that? Because when we need to cut or visualize something, we need an image that starts from the left upper corner with a specific width and a specific height. And we are going to understand what happens or what is going to happen if we missed that part. So here, for each x and y in the x and y coordinates, we have subtracted the minimum x from all x's and the minimum y from all y's, and called it region x shifted and region y shifted. And to construct a polygon and fill it with a specific color like white using the fill poly or fill polygon in the OpenCV package, we need to pass the x and y as list of tuples. To do that, we use the function zip, and by passing the shifted x and shifted y, it will take each element in this list and the corresponding element in this list and combine them together into a tuple called x and y. By doing that using list comprehension, we can get a list of tuples that include the shifted x's and shifted y's. Then this function also need the values that pass to it to be integer. So we have converted using the NumPy package from a list of tuples to an array with a type of NumPy integer 32. In short, we have converted that into NumPy array with a type integer. We did that because, as mentioned, this function requires all polygons or all coordinates to be integers. So we have created a black mask with a specific width and height. Then we filled that mask using the coordinates with white, and white here in the grayscale is 255. And then to display it and deal with that mask as an image, we need to convert that mask to unsigned integer 8. So we used the NumPy function as type to convert it to uint8. So this now is an image with a grayscale from 0 to 255, which contains the mask that should be multiplied later by the region image to get the region of interest. By displaying the region mask, and we write here using the mshow function, and we write here the map is gray because we are dealing with a gray mask or a gray image, and then execute the plot.show to display the figure. By executing the code, it will display the mask like that, with the white region that reflect the polygon or the vertices, which contain the benign region, and the black region that should be ignored because it's not benign. So now we have a mask. We need to get the region. So to do that, we can use the read region by defining the top left corner of the x and y coordinates, width and height, and we need to read from level 0, which is high resolution. To multiply both of them together, we need first to handle that region as an image, so we used the function asType to convert it also to unsigned integer 8 to be similar to that, so we can later multiply both of them together. And if you remember, when we said there are 4 values for each pixel, the first 3 are RGB and the fourth one is the alpha, we need here to get rid of the alpha channel, so we can use the function cvt color or convert to color in the OpenCV package by passing the image and using the attribute or the definition RGBA to RGB. So that one now will have only 3 channels and its type will be unsigned integer 8. We have also the mask with the type unsigned integer 8 by multiplying both of them using the bitwise, and in the OpenCV package we can get the region of interest. The first 2 attributes will be the regions, and we will define the mask to be the region mask. And here we confirm that the ROI is only containing 3 channels by using the same function. Then we made a subplot with 3 columns, the first for the mask, second for the region, and the third for the ROI. If we executed that code, it will get something like that. We have a mask, we have a region. By multiplying both of them, only the region of interest will be displayed. And when we scroll here or here, we will find that each pixel only contains 3 values instead of 4 like the previous code. So in that code, we also compiled the first 2 codes by reading the whole slide image and the annotations, combined them together and got the annotation list. And based on the annotation list, we can also change the index here to be, for example, 2 to get another region. By executing the code, it will display something like that. This is the region mask, this is the image. By multiplying also both of them, we will get the region of interest. And to understand why the shift to the left upper corner is important, we have commented this part and repeated it again without the shift part, just keeping the XY as it is. And instead of using the width and height, which is calculated here, we have only used the maximum of y and the maximum of x without this portion, which is also considered the shift. And then we displayed the mask and the image, and we have commented the bitwise and between both of them, because if we kept it, it will raise an error. And we only displayed here the mask and the image. By executing the code, it will produce these 2 images, the mask, which is placed in its original place, and the region image, which we cutted from the left upper corner with a specific width and height using the red region image, as we did here in this line. So we had to shift each point to the left upper corner so it can map the same coordinates of the original region image. And here don't say that this polygon is reduced in size. Maybe this is done because of the visualization. If you went at this point, you will find that the width and the height of this image is about 5K by 7K, while here is only 1.7K by 3.3K. So if we zoomed this region, it will display the same sizes, width and height for both of them. So originally the mask is not reduced in size, but because of the visualization, it appeared like that. So we wanted to convert and shift from its original place to be similar to the region image so we can apply masking between both of them. So in that lecture, we were able to read the whole slide image, read the annotations, explore the different tags and the hierarchy of the XML file, merging both of them to draw the annotation on the whole slide image using the thumbnails, and finally we could extract a specific region based on a specific mask multiplied by the region image to get the region of interest, so we can later use that region of interest to extract patches and tiles.